In this video lesson, we'll be looking at the structure and the property of proteins, and we'll be building on our knowledge from what we learned about proteins in our biology course. So, amino acids are the building blocks of proteins, and they consist of a central carbon, so that's this one right here, that's bonded to both an amino group right here and a carboxyl group right here. And then there's also an R group here, and that's the thing that makes all the 20 amino acids differ from one another. The simplest one is glycine, and it has hydrogen for its side chain. And they have different properties depending on that R group. So this R group could be polar, it could be non-polar, it could be ionic, it could be acidic or basic, and so on. So all of those things will affect how proteins interact and behave. So there is an exam tip from our textbook that's helpful. So first of all, the 20 amino acids and their structures are found in section 33 in our data booklets. You don't have to memorize them, but you do need to be able to identify the functional groups and predict properties, whether you think it will be at this amino acid will be polar or non-polar, and determine intermolecular forces and whether or not they may be acids or bases as well. So as I mentioned before, glycine is the simplest amino acid, so its R group is hydrogen, which means that it is not a chiral carbon, that central carbon, because there are two hydrogens. It doesn't have four things attached to it. So all the other amino acids, however, have a central chiral carbon, which then makes them optically active. And again, there'll be two different configurations. So to determine whether the amino acid is L for left or D for right, we use something called the Korn rule. So if the carboxyl, the R group, and the amino group are clockwise, they're the D enantiomer. And if they're counterclockwise, they're the L enantiomer. So if you look here, this is the L configuration because it goes carboxyl, and then the R group is here, and then the amino group, so it's going counterclockwise. So that's L. And then the other one is the D because they are going clockwise. So just a note, most amino acids that are built naturally, so within plants or animals, they're found in the L configuration, so they're the counterclockwise model, so it goes carboxyl, R group amino, counterclockwise. There are a few instances where there's some free amino acids that will be the D enantiomer, so then they'll go clockwise. However, they're found in the cell walls of bacteria and the venom of certain animals, so they tend to be toxic for some reason. So because amino acids have an amino group and a carboxyl group, they have the ability to act as both an acid or a base. So the carboxyl group is acidic, hence carboxylic acid, and it can act as a proton donor. The amino group is basic, so it can act as a proton acceptor. So you can see that happening here. So if the pH rises, so this is our neutral pH where it's the carboxyl and the amino, no hydrogens have been donated or accepted. If the pH goes up, it will act as the acid and it'll donate a proton. So it'll lose a proton. So the carboxyl will have one less. And then over here on the right, if the pH falls, it'll start accepting protons. So it'll be acting as a base. So there's a new term called Zwitterions. So amino acids have the ability, or they are Zwitterions, because they can both have a positive and a negative charge at the same time. So if you look in this example, this amino group, it's accepting a proton, and then here, this proton is releasing, giving its electrons there and leaving. It's being taken up by an amine group. And so the Zwitterion actually has a positive charge at one end and a negative charge at the other. So in neutral situations, amino acids are acting as their zwitterions, so they have the positive and the negative end. When it gets very acidic, they'll act as a base, and the carboxyl group will accept that extra hydrogen or the proton, and so it'll become a cation because it's only positively charged. And if very basic environments occur, they act as acids, and so they will donate their proton right here to become an anion. So the graph up below shows you the pHs where that will change, and this will vary depending on the amino acid, but the zwitterion is in large quantities. It's mainly the only ion in neutral and slightly basic and slightly acidic parts, and then it pretty much only exists as their cation in the acidic situations, highly acidic, and then highly basic, they only exist as their anion. So every single amino acid will have a different isoelectric point, and that is the pH where the zwitterion is formed. So this will vary based on the R group of the amino acid. It's also the isoelectric point is symbolized as lowercase pi. Here's a few examples. So for alanine, the R group is methyl. So it's nonpolar, and its isoelectric point, or pi, is 6.0. Serine is polar. It has a hydroxyl. So it's polar, and its isoelectric point is lower. Glutamic acid is polar and acidic because there's a carboxyl group there. So it has a very much lower isoelectric point. And then lysine is polar and basic, so its isoelectric point is higher than the neutral point. So there's another exam tip found here. So section 33 in the data booklet, same one where you find the amino acids. So it has the name, the 3-letter code, and the structural formula, and the isoelectric point for all 20 of the amino acids. But the structural formula doesn't show you the zwitterion form, so you need to be able to change the standard form of the amino acid to the zwitterion form. And that's simply to take off the hydrogen from the carboxyl and stick it onto the amino group. So let's have a look at melting points. Because they exist as zwitterions, amino acids have high melting points, so between 200 and 300 degrees. The R group will impact the melting point, and amino acids are crystalline solids. Again, because they're zwitterions, they're soluble. The R group will also impact their solubility, so it'll determine how soluble they are. And because amino acids are amphoteric, they can actually be used as buffers themselves because they can either absorb a proton or donate a proton, depending on the situation. So if they're in an acidic environment, the amino acids will accept the proton and make them more positively charged. And then if they are in a basic environment, they will lose a proton and become negatively charged. So they actually will affect the pH, and they'll buffer that solution. So if you recall from last year when we learned about acids and bases, we learned how to use the Henderson-Hasselbalch equation. So pH is equal to the pKa plus the log of the concentration of the base over its conjugate acid. So a student is making this is our example from our textbook. A student is making a solution of simulated gastric juice in the stomach by preparing a buffer for an experiment. If 0.02 liters of 0.1 mole per liter carbonic acid is added to 0.01 liters of 0.1 mole per liter sodium hydrogen carbonate, what is the pH of the buffer solution? So the first thing we need to do is find our total volume. So we're adding two volumes together, so that would be equal to the 0.02 plus the 0.0100. So we have a total volume of 0.0300 decimeters cubed. So the next thing we need to do is find the concentration of carbonic and sodium hydrogen carbonate. So carbonic acid is, because we were given the volume and the concentration of them initially, we need to times the volume by the concentration initially and then divide it by the final concentration. So it's going to be equal to 0.0200 decimeters cubed times 0.1 mole per decimeter cube divided by the new total volume. So that's going to be equivalent to 0.0667 moles per decimeter cubed. And then we do the same thing for sodium hydrogen carbonate. So this time we only had this time 0.01 decimeters cubed, but it was the same concentration. And then we divide it by the total volume. And our answer is 0.0333 moles per decimeter cubed. So now we have our concentrations of the acids and bases. So basically we used our dilution formula rearranged. So remember, dilution formula is C1V1 equals C2V2, and you just rearrange to find out what your final concentration was. So all we did was we multiplied this initial concentration by the initial volume, and then to find the final concentration, we divided it by the final volume. It was like doing a dilution because we mixed it with another volume. So now we can plug our information into our Henderson-Hasselbalch equation. So first you have to look up the pKa for carbonic acid, and we can find that in the data booklet, and that's equivalent to 6.35. And then we're going to add the log of the base over the acid. So it is 0.0333 over 0.0667. And I'm just going to extend down here. So that's going to be 6.35 plus the log of 0.5. Oops, I don't know why that happened. And that's equal to 6.35 plus negative 0.301. So that's equal to 6.05. So the pH is 6.05. So now let's have a look at polypeptides. So polypeptides are when amino acids are joined together. They're joined together through condensation reactions, and the bonds are called peptide bonds. And they're always added with the amino group to the left and the carboxyl group to the right. So the N with the amino group is called the N terminal for amino, And then the end with the carboxyl group is called the C terminal, the C for carboxyl. So you can see how the amino acids are different. You can see at each of the R groups that they do differ. And then, so this is the structural formula, and this is their 3-letter abbreviations. So cysteine, arginine, valine, and so on. Now, proteins or polypeptides can undergo a hydrolysis reactions. So they can be digested by enzymes. When this occurs, water has to be added back across the peptide bond because remember, condensation reactions, water is removed to condense them together. So that's how you separate amino acids, by undergoing hydrolysis reactions. So now we'll review the 4 levels of structure, which we did in biology class. So the primary structure is the amino acid sequence, and the amino acids are held together by covalent bonds, also known as peptide bonds. So there's our example. So it's the simplest structure, so it's just the simple sequence, but they won't stay that way because the side chains and the aminos and the carboxyls are going to want to interact with one another. So the next stage of protein structure is secondary structure, and that's when the polypeptide will either fold or coil. And what's happening are the carboxyl and the amino groups of the nearby amino acids will hydrogen bond with each other. So proteins will have multiple alpha helices and beta pleated sheets throughout the structure. So it won't just be all alpha helix and it won't be all beta pleated sheets. It's going to be a mix of the both, and you can recognize them in diagrams. So if you see coils, then those are alpha helices, and if you see they'll either be represented this way or they may look like just an arrow. Let me just draw an example. Sometimes you see beta sheets represented like so, and then maybe another one going this way. So that is another way of recognizing a beta sheet. So alpha helixes, the polypeptide will coil and it'll keep the backbone on the inside, and the R groups will project outwards. And you can see it a little better from the above view. So the purple part are the R groups pointing outwards. You can see it here too, that they're pointing outwards. And an alpha helix can usually range from about 4 to 40 amino acids in length. The average length is around 10. Beta pleated sheets, on the other hand, are when parts of the polypeptide fold in a pleated manner and they run in opposite directions. And in this case, it allows distant sections of the polypeptide to interact with one another. The next level of protein structure is the tertiary structure. So the polypeptide folds into its 3D globular shape is what it's described as. And this lets it exist in aqueous environments by keeping all the non-polar groups inwards, and the polar sections can stay on the outside. So you can see here, there's some alpha helices, there's some beta sheets, like I represented before. And all the red parts are parts that don't have either of those structures. So they're interacting in their own way by like clumping the polar groups in one section and the non-polar in a different. And then these black lines are structures known as disulfide bridges. So some amino acids have a sulfur in their R group, and they will actually covalent bond within a nearby sulfur in a different amino acid. And they actually form covalent bonds. And those can be broken sometimes. We actually, when we do the process of perming or permanently straightening or curling our hair, we use chemicals that break the disulfide bridges. We reshape the hair and then we reform the disulfide bridges by adding another chemical. And then you have your hair will stay permanently curly until it grows out, or permanently straight until it grows out. So a lot of different interactions are at play in tertiary structures because the R groups are interacting with one another. So you can see here some deeper structures here. So we can have hydrogen bonding. So we can see here that the hydrogen on this one is forming a hydrogen bond with the lone pairs of electrons on this oxygen. We have London forces, so these two are non-polar side chains, but they're being attracted by London forces. Disulfide bridges, so two cysteines have oxidized their SH groups to form a covalent bond. And then over here, here's two charged particles, so two ions, so the NH3 plus and the O minus are interacting with one another. Those ones could be broken by changing the pH. So we can actually have different interactions due to the change in pH that the protein is found in. The final level of protein structure is called quaternary structure, or the fourth degree. So this happens when there's two or more polypeptides joined together. And this is an example of hemoglobin. There's actually 4 here, and they're being held together by intermolecular forces. We'll spend some more time talking about hemoglobin in a later lesson. And this is a final recap of everything, of the 4 levels of structure. So primary is just the sequence of amino acids. Secondary is coiling or folding into alpha helices or beta sheets. Tertiary is folding it up into its 3D structure. It's the side chains are interacting with one another. And then quaternary structures when 2 or more of those coiled polypeptides interact and hydrogen bond together.